Section 20.4G. Explain why cancer is most often a disease of old age. Cancer is a disease caused when cells divide uncontrollably and spread into surrounding tissues. Although cancer has been studied for years, there is yet to be a cure. One of the major correlations found while researching cancer and cures has been age. Aging is a time-dependent deterioration of physiological organ functions needed for survival. It is a substantial risk factor for cancer. The principal connection between aging and cancer is simply time. Aging allows enough time for mutations to accumulate within the body. While aging and cancer may seem to be the inverse of each other, aging cells become less fit when cancer cells gain fitness. Both contribute to the time-dependent accumulation of cell damage. In this video, we will be discussing 3 of the major characteristics shared by aging and cancer: genomic instability, telomere attrition, and epigenetic alterations. Genomic instability. Genomic instability is one of the most common marks that is seen in both cancer and aging. Free radicals are especially reactive atoms that have unpaired electrons and can be produced either by natural biological processes or from outside pollutants and toxins. We are constantly exposed to external radiation and free radicals that can damage cells, proteins, and our DNA. While most of these mutagens are harmless and can be fixed by our DNA repair systems, those that are not can be very destructive. Genomic instability is defined by a high frequency of mutations within the genome. It is a distinguishing factor of almost every human cancer and is the primary enabler to many of cancer's typical traits like sufficiency in growth signals and increased metastatic potential, which is a tumor's tendency to form a second metastatic lesion and spread. Mutations in the DNA repair system can induce hereditary cancers by giving rise to increased mutational rate and genomic instability. These mutations can also cause premature aging or progeroid syndromes, which are quite prevalent. Genomic instability and mutations lead to aging with small point mutations or deletions and large translocations. This can be caused by the changes in regulatory sequences that will lead to a progressive decline of organ function due to alterations of the proteome and homeostasis. And less commonly, genomic instability can cause changes in the sequences of protein coding, which leads to disturbances of the phenotype in somatic cells. One of the most effective ways to prevent cancer and aging is by regulating and maintaining genomic stability.
Telomere attrition. Most normal human cells have a limited lifetime. Afterward, they undergo controlled cell death called apoptosis. Cells can only undergo a limited number of cell divisions before the cap end of the DNA is used up and the cell enters apoptosis. Short telomere lengths have been associated with many age-related diseases. The telomere function is disturbed as the body ages. The enzyme, human telomerase reverse transcriptase, or HTERT, has the ability to build back up on the cap ends of the DNA that otherwise get shortened by each cell division. Once this telomerase becomes unregulated, cancer cells gain a limitless replicative growth. In 80 to 90% of fully malignant human tumors, the telomerase is unregulated compared with normal tissue. By avoiding shortening cap ends, the cells are never able to enter apoptosis. Multiple studies following telomere attrition have shown it is a big risk factor for cancer development. So, HTERT may in some cases act as a risk factor for developing cancer, while in others it acts as a driver for the aggressiveness of cancer. Next is epigenetic alterations. Epigenetics are changes in gene expression without altering the DNA sequence. Methylation is a form of this. DNA methylation has been thought of as the predictor of human age in many studies. It is a common mechanism of gene regulation. Promoter region hypermethylation is associated with gene silencing, while hypomethylation is associated with increased gene transcription. Cancer cells are broadly characterized by this hypermethylation and the silencing of tumor-suppressing genes included in cell cycle regulators. They are also associated with hypomethylation, as it seems to have a role in causing oncogenesis by causing instability in chromosomes. Next is histone modification. Histone modification has been linked to both aging and cancer. Histones can be altered by acetylations, methylations, sumoylations, ADP, ribosylation, ubiquitination, and phosphorylations. These alterations cause the electric charge of amino residue on histones to change and make them bind negatively charged DNA strands tighter or looser together. This results in actively transcribed loose euchromatin or the tighter silenced heterochromatin. These epigenetic changes also work in the recruitment of the transcription regulation machinery. The modifications occur on the histone tails and are controlled by numerous enzymes. Analysis of large sets of sequenced DNA from tumors reveal that histone methyltransferase are frequently mutated in cancer. Aging is also associated with the specific complex alterations of histone and chromatin distribution. The chance of all these alterations and mutations increase with age, showing that as we get older, the chances of gaining cancer increases.